Good afternoon. This is Wednesday. Forgot the date a second. This is Wednesday, October 21st. I'm getting to the follow-up on the protein folding model a little late. I apologize, but this is the second module for the modeling kit for protein folding. Last time we talked about the tertiary level, the third level of protein folding. This focuses on the second level of folding, the secondary level. So primary is what you get right after transcription by the ribosome. Right after transcription from the ribosome, you have the backbone. You have the primary structure. This is representing that repeated chain of amino acids, that sequence of amine group, central carbon, carboxyl group, amine group, central carbon, carboxyl group, amine group, central carbon, carboxyl group, amine group, central carbon, carboxyl group. That repeats all the way down the backbone, all the way back down the backbone. On the last time, we added on these clips, which represent the bond with the central carbon to the side chain or R group. And when you put in different side chains or different R groups, that's what causes chemical interactions, which start causing the protein to fold and take on its three-dimensional shape, which determines its function. What you may not have noticed being at home is when we did this part, the models themselves were fairly flimsy. There was a lot of give to them, a lot of wiggle to them, which happened because we skipped on purpose. We skipped the secondary level of folding. Secondary level of folding is all about adding stability to the model. There's two basic types. There is a beta pleated sheet where literally this protein, and I know I'm blocking the view for most of it, this protein folds into a series of pleats. And what holds us together are hydrogen bonds. So we have these white clips, which are going to represent the hydrogen bonds. What's not involved here is the R groups or the side chains. The R groups or the side chains have nothing to do with the secondary structure. So this represents a hydrogen bond between the carboxyl or amine group of one amino acid and the carboxyl group and amine group of a nearby amino acid. That's a hydrogen bond. This might be from the carboxyl group on this amino acid to the amine group on this amino acid. So these are bonds between amine groups and carboxyl groups of the primary chain. This is not in any way involving the R group or side chain. So you can see if I start adding some of these clips in as hydrogen bonds, this is really going to start to strengthen this molecule up. All right. And there's not a whole lot of give there. On this end here, I got a lot of give because I don't have the hydrogen bonds in place. But over here, there's no give. All about stability. And this pleated sheet, in general, the pleated sheets are used in structural proteins. They provide a lot of strength. Muscle would be a good example of a pleated protein, very strong sheets of protein. Besides the beta pleated sheet, we can also have an alpha helix. In an alpha helix, what happens is it forms a spiral. And with the spiral, again, you're going to have these bonds between carboxyl and amine groups, and that is going to start to stabilize that. Hopefully I'm not moving too quickly that you can't see what's happening. And this starts to build more of a spiral or coil-like structure. That's an alpha helix. This is more of what we call a globular protein. These are proteins that float around in the body fluids to carry out jobs. These would be things like enzymes, antibodies, hormones. Tend to be more globular. That tends to be, in general, simplifying, but generally is involving globular proteins, which are alpha helixes, where structural proteins often incorporate a lot of beta pleated sheets. And a lot of proteins actually incorporate both. So in the activity, if you look through the pages, we are creating a structure known as a zinc finger, just as an example. And it talks about it on pages 1 and 2. On page 2, I'm looking at the packet. You want to probably look at the handout as well. Page 2, it gives me another sequence of R groups or side chains to add to my molecule and the distances to put them. All right. And then it talks about folding and making beta pleated sheets and alpha helixes. Here's the alpha helix part. And ultimately, when we do all that, we end up with a structure that looks something like this. So I'm speeding ahead. This is one that a group made during class today. Okay. And so you can see over here, I've got my beta pleated sheet. And on this end, I've actually got an alpha helix. This is a true structure. This is a lot more stable and a lot firmer than the model I made the first time with just the tertiary structure. So this really does give the molecule some structure. The orientation of these R groups is very important for the zinc finger to function. The zinc finger literally binds to zinc. So if I met this blue ball be zinc, oops, excuse me, this would bind here, and that would even stabilize the molecule even further. And this allows this to do its job. So in the questions for this, there's not very many. I think there's like 5 questions. It asks, question 1, both alpha helixes and beta sheets are stabilized by hydrogen bonds. Which atoms share the hydrogen in these weak bonds? The atoms that are sharing the hydrogens are the amine groups and carboxyl groups of the primary structure, not the side chains of the R groups. It's the amine groups and carboxyl groups of the primary chain. That's what's sharing it. So it's coming from the backbone atoms, not the side chain atoms. Very important. In the zinc finger, there's 2 secondary structures. On the one end, there's the beta pleated sheet, which has the 2 cysteines, the 2 green clips. And then on this side, there's the alpha helix, which has the 2 histidines. And the cysteines and histidines sort of form a clamp, which then traps the zinc so that it can do its function. So the zinc atom binds to the cysteines and histidines and helps stabilize this so it's not flexing like this. It locks it in so it can do its job. Why will the zinc finger bind to DNA? Because that's what it's involved in working with. Arginine on here, arginine has a positive charge. Well, DNA always has a negative charge because of the phosphate group. Remember, DNA nucleotides always have a phosphate group. Phosphate groups are negative. So the positive arginine will attract the negative DNA. And the zinc finger is very much involved in allowing us to do some key jobs in terms of DNA synthesis. Okay. So it's really, I think, all I need to show you for that. I mean, I would read through the packet and I think it'll make a little bit more sense. But the takeaways, the big takeaways here is the secondary structure provides stability. Secondary structure provides much more stability, lots of strength to the protein structure. There's 2 types, beta pleated sheets, alpha helixes. Beta pleated sheets tend to be more structural. Alpha helixes tend to be more functional, globular proteins. And the other really, really key thing is the secondary structure is only impacted by hydrogens on either the carboxyl or amine groups that are attached to the central carbon. These side chains These R groups have no role in secondary folding. They get involved at the tertiary level, which is what makes the molecule functional. Hope that makes some sense. And this will allow you to do what you need to do. All right, that's all I got. Have a good one.